law theory is also known as the law of segregation. He predicted meiosis 1 100 years before it was even known, that the members of each allele separate when gametes are formed. We just studied this in the last unit about meiosis. In meiosis 1, homologous chromosomes separate and we get the reduction number. Here's a quick review of that. We've got a red and a dominant red and a recessive blue trait here on two homologous chromosomes. In meiosis 1, they separate and both chromatids go one way and the other, the recessive ones go to the other way. So now we're going to form gametes. And so we have 50% of the gametes are dominant and 50% of the gametes are recessive. So a monocross showing capital white and lowercase for blue. It's a cross between two truebreds. So we're going to start with capital W crossing with homozygous recessive little w, which would be blue. Again, this is the way Mendel did his classic experiment. The dominant homozygous dominant versus homozygous recessive. So if we set these up on the Punnett square, we've got from the dominant a capital W and a capital W will be the possible gametes that the one parent can give. It's all they can give because they're big W, big W. Then we have the other parent, the blue individual. The blue parent can give a little w and a little w. So the gametes are set up on the outside. And then big little is one possible offspring if those two gametes, if those two sex cells get together, or if these two get together, it would be, again, the same thing. And a third and a fourth, all identical. It's the only possible outcome. The F1 generation will all be heterozygotes. We have the same thing again, and now the genotype would be 4 heterozygotes. They're all heterozygotes. And the phenotype, all white. Now, if we cross the two of these individuals that are inside the box, these are the F1s. If we cross two of those or let them self-pollinate, same thing, we're going to get a parent that's a big W little w crossing with a big W little w. So here we have the F, a cross between two F1 generations or a self-pollinate. They're both heterozygotes, so we're going to get a big W and a little w, and a big W and a little w from the other parent. Only possible gametes that those parents can make. And the first offspring would be capital, homozygous white. Second one, big W, little w, heterozygous white. Third one, same as the second, heterozygous white. And the fourth one, little w, homozygous recessive, blue. So genotypes, fill them all in again. We're going to get the genotype. What genes are actually in that box? 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. Phenotypes, a little bit different. 3 whites and 1 blue.